Hi, everyone. Thank you for joining us today. I just wanted to go over some quick housekeeping items. In order to go ahead and connect to our audio, you can utilize your computer's audio, or if you would like to use a call-in number, that call-in number information is right there on your first screen. Our panelists will be taking questions throughout the webinar, and so if you do have a question, please feel free to use the Q&A function at the lower right-hand side. And if you are having any difficulties throughout the webinar, you can also use the chat function and message the host, and I will be able to help troubleshoot any of that information. And for those of you who are joining us and having to jump off early or are coming onto our webinar late, we will be recording this webinar and will be provided to Softbones over the next couple days, and they will be able to distribute to those who are jumping on late or jumping off early at a later time. So with that, I'm going to go ahead and pass it over to Deborah Fowler to go ahead and kick us off.
Hello, everybody, and thanks for joining us tonight. I know that it's difficult to find a time that works for all the time zones, but we're excited that we had a good RSVP rate. So we're excited to kick off HPP Awareness Week with this webinar that addresses one of the areas where we get the most questions, and that is genetics. So today we bring you part one in a series on hypophosphatasia genetics. We're calling this one Genetics Boot Camp. I know there's a lot of newly diagnosed people on the line and people who have been living with this disease for a long time, so it may be some information you're familiar with. But the next webinar will be a little bit more high-level science, and we'll dive into the genetics more specifically, where we'll start looking at questions like, how can two people with the exact same mutation have completely different forms of disease? So stay tuned for that. But before we get started, I wanted to quickly provide an overview of what is soft bones. Some of these faces may look familiar to you, and as you know, we're a patient community, and a large part of our communications are on Facebook. But our mission is much bigger than that. We're much more than a Facebook page. Softbones is actually an official 501c3 nonprofit organization, and this is our mission statement here. We provide information, education, and support for people living with hypophosphatasia, and also to their family and caregivers. But a big piece of our work is around promoting research of this rare disease through awareness and fundraising efforts. And so, with that, we also support policy initiatives. We fund grants for research and studies to better understand patients with HPP. And we help patients create their own care teams by finding doctors who have experience with HPP and providing them with education so that patients are better informed to make decisions about their health. So we do a lot of things that you may not be aware of behind the scenes. In addition to our Facebook, we also have started breaking out into regions. So earlier this year, we divided the country up into 8 regions. Each one of them has several region leads that are helping to bring the support down into the communities where you live. If you don't know who your region lead is, please feel free to reach out to myself or Sue or Denise, and we can connect you to whoever your region lead is and make sure that you're aware of any local HPP events that are happening near you. One of the most important ways we support research is through our patient registry. In partnership with Sanford Research, we created a patient contact registry where basically anybody who's interested in participating in any sort of research can opt in to being contacted by researchers. This is really important for carriers as well, because we're looking to learn more about why people who are carriers are starting to exhibit symptoms. So if you haven't already put your information into the registry, make sure you do so. We've already had a neurologist using it to conduct a study to better understand how HPP impacts cognitive function. So it's very important, a very, very important piece of what we do and helps us to fulfill our mission. And then finally, before we get started, of course, tomorrow is officially HPP Awareness Day. We kick off a week of activities, including this webinar, which wouldn't be possible without Global Genes supporting us with this webinar and the infrastructure, so thank you for that. And then obviously the expertise of our genetics experts from Duke. So we're really appreciative that they're taking the time to be with us this weekend. We'd also like to announce the topic of the next webinar, which is going to be on vitamin B6 and HPP with Dr. Eric Rush, which will be in November, date TBD. Tomorrow is obviously the big day. A reminder to wear your HPP t-shirt and post it along with the hashtags HPP Aware and HPP Day 2018. Also this week, look in your mail for our first annual engagement survey. We will be conducting this survey hopefully on a regular basis annually so that we can continue to gauge the areas of support, where you want to have more information, what topics are of interest, and that'll help us to focus our efforts in terms of the services that we provide. And then also there will be a Stella and Dot fundraiser that's going on this full week, held by Christy McKillop, who's a fellow HPP mom, and she's generously donating all of the proceeds from the online fundraiser to Soft Bones. And then you may see some pictures. There's a local spin-a-thon here in New Jersey happening where people are cycling and raising $1 for each mile for HPP. So with that, without further ado, I want to introduce our speakers tonight. We have two experts on the line with us. One of them is Stephanie DeArmey, who's a physician assistant. She's worked with the Duke Metabolic Genetics Group for 18 years. She focuses on both research and clinical care for patients with rare genetic disorders and enzyme replacement treatments. And she's joined by Lauren Flueckinger, who's a certified genetic counselor at Duke University Medical Center in the Department of Pediatrics. She began her professional career at Duke in June and works in both clinical and research roles, managing approximately 450 patients, serving as a clinical research coordinator for multiple research studies at Duke since 2013. She has particular interest in lysosomal storage disorders, glycogen storage disorders, and of course, hypophosphatasia. So with that, Lauren and Stephanie.
Can you hear me?
We can.
Okay, great. Thank you so much to Soft Bones for having us here. So this is our first time doing such a webinar, and hopefully it's helpful for those of you out there. To start with, Lauren and I work very closely together, and I think having a clinician and genetic counselor is a good team when dealing with a disease such as HPP. So we have some objectives listed here. I'm going to tell you, discuss a little bit about the disease, and it helps to have an understanding of the disease to know why it's important to understand really the genetics of the disease. So we'll move on. So what is hypophosphatasia? Those of you on the line and on the call probably have a good understanding of what HPP is if you're a patient or a family member or a loved one. But it is a rare inherited metabolic disease, and it's caused by mutations in the ALPL gene. So that's important to understand that it's an inherited disorder. The mutations in this gene, it affects the tissue nonspecific alkaline phosphatase isoenzyme. And we know that this enzyme is important for bone and teeth mineralization. And we know that without it working properly, it leads to substrate accumulation. And we'll talk a little bit about that here in a minute. And as you talk more about the disease, you'll often hear tissue nonspecific ALP as well as ALP, and they can essentially be used interchangeably. So the mechanism of the disease, essentially you start with that mutation, and then that leads to the deficiency in the ALP activity. And then from there, you have some substrates that build up in your body. And those of you that have seen clinicians may have been tested for some of these. Inorganic pyrophosphate, or PPI, pyridoxal 5-phosphate, or PLP, also known as the circulating form of vitamin B6, or phosphoethanolamine, PEA, which is found in your urine. When these substrates accumulate, it affects the metabolic, affects your body system in a lot of different ways. That's a whole talk in itself. I'm not going to go through this, but what you see from those metabolic changes are clinical sequelae in multi-systems, not just bone, not just teeth, but it can, of course, be bones with rickets and osteomalacia. You also see seizures. You have respiratory issues, increased calcium and phosphate, as well as deposition of calcium in your kidneys. So the prevalence of HPP is really not known. We're learning about it, and things like the registry that Softbones has is a very important way to learn more about who has HPP. But what's been reported is the prevalence, the global prevalence of severe HPP. It ranges from 1 in 100,000 to 1 in 300,000, so quite rare. And we also know from such things as these registries that specific genetic mutations are found in some populations. And here we have some that are reported, including those in Canada, Japan, Europe, as well as the U.S. HPP is a continuum of disease. And what I mean by that is that it's found from the perinatal form, period of life, all the way to adulthood. The severity varies. We know that in perinatal, the infantile form, it's typically more severe in the presentation. And then it goes all the way to the odonto form, which by definition is only includes symptoms regarding the teeth and no other systemic symptoms. So this progressive clinical sequelae, essentially the cumulative effects of the loss of this ALP function, it leads to the sequelae and impacts the skeleton, but we know it impacts other organ systems as well. So these potentially serious and life-threatening consequences are particularly found in the perinatal period, as I mentioned. Significant mortality, almost 100% in patients who have symptoms that start in utero or at birth, and then approximately 50% mortality in patients who have symptoms shortly after birth. But regardless of when the symptoms start, we know that it's life-altering as well as debilitating, and it really impacts lives. So I have, I don't know, I had two slides here, and they sort of were stacked on top of each other. So there was a slide before this that's not showing up. It's the infantile form of HPP, but essentially it's you have the different systems listed here, and it would basically be more severe for the infantile forms, but you have the different systems. So skeletal-wise, you have fractures. In the adults, they're often recurrent, often non-healing, osteomalacia. Chronic bone pain is often a symptom that we see in patients in our clinic, arthritis as well as pseudogout. From the kidney perspective, we often we'll ask for ultrasounds of the kidneys, and we're really looking for any calcium depositions, and they can lead to some renal failure. From a muscular perspective, if you go to some clinicians, they may ask for a good physical therapy evaluation, and we're trying to understand more about how the muscular system is impacted by HPP. But we know that severe myopathy, weakness, the use of assistive equipment, as well as pain is very prevalent. And then dental, of course, the premature loss of teeth. As a patient with symptoms that may be HPP, the differential diagnosis of the list of possible things you might have is quite big, and here it lists out some of those. And it's actually growing. I think we've learned a lot that these rheumatological diseases, which are listed on the right of the screen, we've found that a lot in a lot of our patients. And so we're learning, again, a lot of the multi-system effects of this disease, and it's important to understand what you have so you know how to treat what you have. So the diagnosis would be based both on clinical presentation as well as your lab findings. So as we talked about, understanding and getting a good history on the skeletal history and manifestations, systemic complications that we talked about, a good tooth history for those of you that are adults, and we would ask you a lot about your teeth, and sometimes it's difficult to remember when you lost your teeth, and so oftentimes we ask for input from parents or siblings, but as well as family history of siblings or parents with HPP. All those things are very important in understanding and making a diagnosis. And then key indicators for lab-wise, of course, having a low serum alkaline phosphatase, or ALP, and having that lab compared to the age-specific norms is very important. And we'll talk about that just briefly on the next slide. Increased levels of the substrates that I mentioned, PPI, PLP, and PEA, and as well as a confirmed genetic analysis of mutations in the ALPL gene. So in this graph, this shows the normal ranges, so the low normal ranges as you age. So you see on the bottom of the graph, you start as less than 1 month, and you can see that normal range is in about the 6-year range, and it goes up throughout childhood, peaking around 11, 12 years old, and then it declines. And then as an adult, that normal range goes down to about the 30, 40 range. So this is really important because not all labs use this age-specific normal range. And so you may get an ALP result that looks normal based on what your lab report says, but then if you go back and check these normal ranges, you might actually be in the low range. So in summary, HPP is inherited, the metabolic disease, and it can have a lot of serious consequences. It's not only a skeletal abnormality, it affects a lot of other organ systems that they're listed here. Patients with HPP have serum ALP activity below the age-adjusted lower normal limit. So that's, again, important, that lower limit is based on your age, as well as increased concentrations of the substrates we mentioned, PLP and PEA. And, of course, molecular testing is important to confirm diagnosis. So why is that important? It can suggest clinical phenotype, and it also can identify your the at-risk family members. So with that, I'll hand it over to Lauren.
Hi, everybody. Thank you all for being here, whether it's, the last hour of work or well into dinner time. I think Stephanie gave us a great setup for what HPP is, how it differs a little bit from patient to patient, but the overall review of what we can see and how we come to find a patient with these symptoms and ultimately, hopefully, diagnose them with the correct diagnosis, which in this case, obviously, we're talking about hypophosphatasia. So my part really here is to talk about the genetics. And I really think in order to understand the genetics of HPP, we have to take a step back. So if I kind of can have everybody remember back to biology class in middle school or high school, when we're learning about how the body is made up, we know that the body is made up of trillions and trillions of cells, okay? And so every cell that we have, be it a cell in our bones or our teeth or our kidneys or any of the other tissues that Stephanie talked about, those cells all have what are called chromosomes inside of them. And chromosomes essentially are just packages of DNA, okay? The DNA that is included in a cell is extremely long. I think they say it's something like over a terabyte of information. It's a ton of information. And so in order to package it inside the cell, it has to be wound up very, very tight. And when it gets wound up very tight, it appears like this thread-like structure of a chromosome here. And so if we were to pull that chromosome out, kind of like a ball of yarn, if we were to pull it all the way out and just have a single piece, that's where you would see that kind of classic DNA shape, that double helix that you see on Time magazine and in political cartoons and everywhere else. So that's where we get the shape of DNA. And if we look even closer at the DNA, certain portions of that DNA consist of what we call genes. So there are about 25,000 genes that exist in humans, and so those are all packaged within these different chromosomes. So what I want to kind of bring to your attention is that if you are remembering back to biology, DNA has 4 base pairs. They are adenine, thymine, guanosine, and cytosine. A lot of times those get shortened to just A, T, C, and G. And so this might be sounding familiar because you may have seen some A, T, Cs, and Gs on your genetic testing report. And we're going to talk about why that is and what that means on your report. So We go from the cell to the chromosome, all the way to the DNA, stretching it out, get to the gene. Why is this so important? As you'll see, the example we're going to use is that the gene essentially acts as an instruction manual. And so the gene is what we use, or what the body uses rather, in order to make these important proteins. In the case of HPP, that gene is the ALPL gene, and that gene, or that instruction manual, makes or codes for the protein, in this case, which is that tissue nonspecific alkaline phosphatase, or ALPphos. So that's the protein that's involved in HPP. Okay, so when we think about chromosomes, like I said, these are thread-like structures that are just jam-packed full of DNA. When we look at the non-disease state, not related to HPP, every person has 23 pairs. So if you imagine these are like 23 pairs of shoes, we get one of the shoes, either the left or the right, from our mom, and we get the other one, again, either the left or the right, from dad. So that's why they come in packages. That's why they come in pairs. The first 22 pairs, so pair number 1 all the way through pair number 22, are the same in men and women, okay? So those don't determine biological gender or sex. That last pair, the 23rd pair, are what we call the sex chromosomes. And if we have an X and a Y, that denotes biological sex of being male. And if we have two X's, that denotes biological gender or sex of being female. So the reason I bring this up is that the ALPL gene, which mistakes in that gene result in hypophosphatasia, that gene is located on chromosome number 1, so the one that's circled there. So what this means is that because the ALPL gene is on chromosome number 1, hypophosphatasia affects both males and females at the same rate. So you may have heard of some genetic conditions like Duchenne muscular dystrophy that affects boys more than it does girls. And that has some effect based on where the gene lives. If it's involved heavily or is actually on a sex chromosome, that affects whether or not boys or girls are affected more. That's not the case in HPP because it's on that chromosome number 1, and as we just said, pairs number 1 through 22 are the same regardless of gender or sex. We see that men and women are affected at the same rate. Okay, so if we were to pull out, again, that DNA even a little bit further, this is basically a picture of the entire chromosome number 1. Okay, as you can see, there's lots of different bands. Those bands can represent different genes, and if you see where that yellow arrow is, that's saying it's located at what's called P36.12, that's essentially the genetic address of the ALPL gene. So if you do a Google search and you're trying to learn about the ALPL gene, you might come across a picture like this. And essentially this is just telling you where exactly on chromosome number 1 that ALPL gene is, because it's not the only gene on that chromosome. There are a number of other genes, but this is specifically where the ALPL gene lives on chromosome number 1. Okay, so if we remember back to the first picture, we said we were going to use the DNA as an instruction manual. That's going to be our example for the continuity of this talk. So when I'm talking about the ALPL gene, think of it like an instruction manual. So you go to buy a chair at a store, but it's not one of those furniture stores where the chairs are already put together and you just put it in the back of your car and take it home. It's one of those stores where you pick out a chair and they give you a box, and you have to then put it together. So in that box, you're obviously going to have all of the pieces you need to make the chair, but you're also going to have this instruction manual. So for this example, our ALPL gene, or the gene that can result in HPP, is our instruction manual. And what we're trying to make in this example is a chair. And so the chair is going to represent that tissue nonspecific alkaline phosphatase enzyme. And again, as Stephanie pointed out, when the alkaline phosphatase enzyme is not circulating at the level we want or not working as well as we'd like for it to, that's what causes HPP. Okay, and for some of you who maybe work in science or have dug a little deeper, this is a simplification of how this happens. There are some processes that happen in the cell called transcription and translation that basically get us from the DNA instruction manual, the gene, to the actual production of the enzyme, or in this case, the chair. But for the purpose of this talk, we're going to keep it on the simpler side. Okay, so what happens when there's a mistake that instruction manual. You take out all the pieces, you start trying to follow, and there's a mistake. You're not going to put the chair together as well as you would like, and that very well may result in you not having a functional chair. So that's kind of what this schematic shows, is that in the previous slide, we had our trusty instruction manual, it worked properly, and as a result, we made a functional chair. But if we have a mistake present, that's going to change how we make that chair and very well may result in the chair not working. Okay, with that being said, there are varying levels of mistakes. And so if you have a mistake where the instruction manual doesn't tell you to put 2 of the legs on the chair and you try to sit in that chair, you're going to fall over. That chair is not going to work well as a chair at all. On the other hand, if the mistake in the instruction manual is less severe and you do make closer to the example of what you're trying to make of a chair, but maybe you have to, like in this picture, prop it up with some books, maybe some of the springs are out of whack, you can still kind of use it, but it's not what we would want. So this is somewhat of the explanation for why patients with HPP, the same genetic condition, can have differing severities. So this is an example of how we have, like Stephanie showed, that continuum of disease. They're both caused by the same type of event, a mistake in the instruction manual, a mistake in the gene, often called a mutation, or sometimes it's called a pathogenic disease-causing variant. But those changes in the instructions result in not making the chair properly. But there can be variability in how improperly the chair is made, and that there is essentially the spectrum you see within patients who all have the same condition but have varying severities. So when we think about types of genetic changes, everybody clearly has differences between each other. That's what makes us individual people. We obviously have genetic differences from, other types of organisms. So how do we know which genetic changes are okay and which genetic changes aren't okay? And so this is some of the terminology that you'll see in your genetic testing report. You might see it in some of your clinical notes as well. And so we're going to talk about the 2 bookends. I'm going to talk about the non-disease state changes, and then I'm going to talk about the disease, and then we're going to talk about what kind of falls in that gray area in the middle. So the very top is a term called polymorphism. Now, polymorphism basically means many forms, okay? And a lot of times you'll hear this called as a variant. You won't hear a polymorphism called a mutation. You'll just hear it called a polymorphism, or you'll hear it called a polymorphism variant. And again, it just means many forms. So this basically means that everything is working exactly like we want it to, but we can have some variation. So all of the chairs are working the way that we want, but maybe the chairs are different colors. We can sit in every single one of them. They don't cause disease. They don't fall over when you try to sit in them, but they are different. So these are things like differences in height, or differences in skin color, or hair color, or eye color. Color seems to be a pretty good example for this. And so how do we decide what qualifies as a polymorphism? And so what has been proposed in the genetics community for a long time is that if there's a specific genetic change that is present in at least 1% of the population, so 1 person out of every 100 on average has this change, then that's a high enough rate of that specific genetic change that we don't feel it's causing disease. As Stephanie mentioned, HPP in the severe forms is affecting 1 in 100,000 to 1 in 300,000. So if we were looking at a polymorphism, that would be 1 person out of every 100. Well, we know that 1 person out of every 100 does not have HPP. So these are the types of just general variation that make people exactly who they are. So it's just person-to-person variation. On the absolute flip side, when we're talking about a pathogenic, which just means causes disease, variant, or the terminology you may hear as well as a mutation, these are genetic changes that are only seen in patients who have disease. In this case, specific changes in the ALP gene that cause hypophosphatasia. So we know that these genetic changes are the cause of the signs and symptoms of that particular disease. So in this case, Perhaps it's tooth loss, perhaps it's a low ALPL, maybe it's a high B6, all of those things that Stephanie talked about. We know that the genetic changes that fall into this category are the reason that that chair is not being built and that we see the downstream effects in the different body systems. So when we find one of these types of changes in a person through genetic testing, that's enough to give someone a diagnosis of, in this case, HPP. And again, we've talked a little bit about the variability that we can see, even though all of these patients technically fall under the umbrella of having a diagnosis of HPP, we do see some variability. So again, that green chair with the books propping it up versus the chair that you can't sit in because it's missing a leg. So what happens when we find something, but we can't really put it into either category? And that's where we hear the term variant of uncertain significance. And this is something you can see on your genetic testing report. And basically, it means what it says, is that we found something, but we don't have enough information right now to say, you know what, it should go in the polymorphism category, or, you know what, we think this causes disease and we need to call it a pathogenic variant. And there's a couple of reasons that that could be the case. Maybe they have seen this in a couple of patients with disease, but they don't know if that's what's causing the disease or if it's not because they didn't find anything else. On the flip side, maybe they've never seen it in the general healthy population, and because of that, they're resistant or leery to determine that it's in one of these categories or not. A lot of times it basically boils down to we just don't have enough data to make a good call. And when in doubt, scientists, if they don't have the data, they don't like to make a call, which can be really hard on the side of the patient who's saying, well, you did my test, you did find something, why can't you tell me what it means? It can be incredibly frustrating. The beauty of science and genetics is that as we learn more, as we collect more data from things like that registry, from studies that you find on clinicaltrials.gov, from clinicians who look at their patient population and write papers so that other clinicians can read them, over time, they get more and more data. And sometimes those variants of uncertain significance down the line with more information can be reclassified. So if your provider ever says to you, you know what, we found a variant of uncertain significance. Some people call them VUSs or VUSs. But in a couple of years, I want to resend this test. It's not because they're expecting your genetics to change. The genetic change will still be there. It's in all of the cells of your body. We don't expect that to change. What we might expect to change, though, is the interpretation that we get and the category that the scientists reviewing that test put that specific genetic change in. And that can be a big deal for folks. If you go from having a VUS to a polymorphism, then maybe you don't need to be getting renal ultrasounds regularly. On the flip side, if you go from having a VUS to having a confirmed known disease-causing variant, that can help with coverage for getting those renal ultrasounds or coverage for treatment. All sorts of different things can be impacted by reclassifying a variant. The other thing I hear people talk a lot about when they look at their testing report is they see that there's a result for sequencing and then there's a result for something called deletion duplication. And there's some confusion about why do we need two tests if we're only looking for HPP, and what do the two tests do that are so different from one another? And this is really where that whole idea of the gene being the instruction manual comes in as a handy example. So if you think of sequencing testing like spell check, and I apologize if that figure is kind of covering up the word spell check, but that's what it says. So if we think of sequencing as reading the instruction manual cover to cover, in this case, in the gene, we're obviously reading those A's, those T's, C's, and G's, and we're looking for any misspellings. That's what sequencing does. If you think about it, if I had a page that had been ripped out of my instruction booklet or a page that had been printed twice, spell check isn't going to find that. It's not going to recognize that. It's only going to be looking for misspellings. And so that's what sequencing does. Deletion duplication, on the other hand, looks for pages, or in this case, genetic information that's either extra, so maybe an extra page in the booklet, or a page that is missing. And so something that won't be picked up by the sequencing test, so we have to do a different type of testing with a different type of technology in order to find those patients who have either a deletion or a duplication in their ALPL gene. The interpretation of both of these tests is the same. So the lab directors and the scientists sit down with what they found, either via spell check or via deletion duplication, and say, okay, what data do we have and how do we classify it and which category do we put it in? Those are the three categories we just saw on the previous slide. So the next thing I hear a lot about is, well, how is HPP inherited? What's the pattern? Do I have a recessive family or a dominant family? And so this is really important because this changes a lot of how HPP tracks through families. It changes the risk to future pregnancies if you have someone who's thinking about having more kids. So it's very, very important. And most genetic conditions play by the rules of only having one inheritance pattern. So you might hear about, say, cystic fibrosis, which is probably one of the best well-known autosomal recessive or recessive conditions. And then you may have heard of things like Marfan syndrome or some types of dwarfism that are dominant. And HPP, unfortunately, doesn't like to play by the rules. And so HPP can actually be inherited as either recessive or dominant. And so we're going to talk about what recessive is and what dominant is. If you're wondering what type of HPP you have in your family, it might help to see the genetic testing reports. It could also help to talk with your clinicians. It could also help to talk with a geneticist or a genetic counselor for those of you who maybe see endocrinologists or rheumatologists, because geneticists and genetic counselors are really trained to specifically understand that portion of things, and they can help clear that up as well. As a general rule, and you'll see this in the upcoming slides, as a general rule, the more severe forms of HPP are seen in this recessive pattern. So the types that are seen during pregnancy or very shortly after birth, and then the more attenuated is a word that we use, basically meaning that it presents later in life. Those families tend to be dominant. But again, it's not a hard, fast rule, so it's important to take a look and talk with your clinician about which pattern your family fits in. So we're going to talk about recessive first. Both of these, take one more step back, sorry. Both of these are autosomal, and again, that just goes back to it being chromosome 1 and being on a body chromosome and not affecting one gender more. So in a recessive condition, it follows the rule of 50% is enough. So again, if we have 2 copies of every gene, we got a copy of ALPL from mom, and we got a copy of ALPL from dad. If one of those 2 copies is working in a recessive pattern, there is no disease. And that's where we get that term carrier. So you may have heard carrier in cystic fibrosis. You may have heard carrier in HPP. And carrier truly means that this is a family with a recessive disease and that one of the ALPL genes is changed or not working, but that person doesn't have symptoms because that backup copy is enough to keep things working properly. So to have recessive disease, both gene copies must be altered in order to be effective with HPP. Now, the difference with dominant disease is that 50% activity rule is no longer in play. It doesn't apply here. So the only way to not have dominant HPP is for both of your ALPL gene copies to be working the way that they should. So if you have one non-working copy, that backup copy cannot put together the muscle to keep things working. So there are no carriers in an autosomal dominant condition, okay? So one change is enough to be affected with the disease and have a diagnosis. Now back to biology a little bit again. So this is for a recessive scenario. So in a recessive scenario, like we said, you're going to have 2 copies. So I used an H for HPP here. So the 3 things that you could be on the right side of the screen, you could be big H, and in this case, that's going to be not a carrier because both of those copies are working the way that we want, and they also don't have hypophosphatasia. On the complete other end, you could have 2 little h's. The 2 little h's are going to mean that neither copy is working, and that's the definition of a recessive disease. So that person with little h would be affected with recessive HPP. And then the remaining are those carriers. So the remaining end up being either big H, or little h, big H. And the reason that there are 2 is because you could get either the big or the little h from either mom or dad. So with 2 parents who are carriers in this state having one working copy, the big H, and one non-working copy, the little h, the way that this comes out for reproductive risks is 25% risk to have a child with autosomal recessive HPP. And it's important to remember that resets. So if you're flipping a coin, what I flipped on the flip just now has no bearing on what I flip next time. So if, they say, oh, it's 25%, well, if I have 4 kids, 1 kid will have HPP, the other 3 won't. That's not the case. Every time, what you're going to flip with the coin has no bearing on what you had the last flip. So it always resets. And as we mentioned, this is typically what we see for patients with those more infantile or severe presentations. So this is the same picture for a dominant disease. So on the right-hand side here, and I apologize, in the recessive form, I used big H as working properly, and here I've used it as not working properly, so I apologize for the confusion, but they stand alone, and if you just look at them as separate examples, they should still make sense. So for this example, for a dominant condition, on the right, these are still the 3 possible outcomes. You could have someone who has big H, which again, in this case, big H is going to be the non-working copy. And those patients would actually have almost kind of what we call a double dominant effect. So they would have even more severe HPP. If you only had 1 non-working copy in dominant disease, remember the backup copy can't fix the barrier. It can't make up for the non-working copy. So if you have 1 non-working copy or a big H here, you have dominant HPP. And then little h here is no disease. So as you guys can see, as opposed to the previous example with recessive HPP where there was a 25% risk, in this case there's a 50% risk because we know that the unaffected parent, the one that's on the side, parent number 2, only has a little h to give. They only have a working copy to give. So essentially what it comes down to is what parent 1, who in this case is affected with dominant HPP, gives. And then it becomes just like the flip of a coin. They're either going to give the working copy or they're going to give the non-working copy, and that's going to come out to 50% dominant HPP and 50% no disease. And this tends to represent more of those childhood, juvenile, more attenuated or kind of later onset presentations. And again, there are no carriers in this scenario. Okay, so the last thing that I really get people talking about is I understand the genetics, I can draw the square like they showed me in clinic, but I can't read my report. It makes no sense to me. So we're going to go through how we can write what we find on a genetics report, and then we're going to go through an example that Stephanie had on one of her previous slides of one of those more well-known, I think it's the European genetic change, and we're going to show you exactly how that would be written. So the short of it is that just as we have the ALPL gene as the instruction manual and the enzyme as the chair that we made, we can either report what was found based on the exact change to the instructions or the DNA change, and that's what we call cDNA nomenclature, and nomenclature is just a really fancy word for saying how we name something. Or we can talk about the exact mistake that occurred in the chair, and that's called protein nomenclature. And so you'll see when they're written out, one has a C in front of it, and that lets you know that this is the cDNA type versus a P, which stands for protein, and then it's going to follow a similar format of telling you where exactly in either the gene or in the protein the change occurs and then what that change is. And this is really best illustrated by an example. So before we show the example, it's important to remember, again, that if we're using the DNA name, we're going to have those A, T, C's, and G's that are present. We're going to basically be talking about what should be there and then what is there. However, when we look at a protein, we expand beyond those 4 letters because proteins are actually made up of amino acids, and there are 20 of those. So as you can see in this chart, there's many more things that we could see, and they actually have their full name, they have a 3-letter code, and they have a 1-letter code. And in fact, C, T, G, and A are all 1-letter codes for some of the amino acids, and that can be confusing. But if it's the protein name, that C, T, G, and A are not standing for those 4 DNA bases that you'll see in the DNA name. And again, this makes more sense when we look at an example. So we're going to take a look at this European common variant, and as you can see, we have a name that starts with a C period, and then we have a name that starts with a P period. So if we look on the left-hand side, this is basically saying that in the ALPL gene, so again, DNA means that we're looking at it in the gene, and if it's causing HPP, then we know that means it's being used or being reported in the ALPL gene, because that's the gene associated with HPP. So this is saying, in the ALPL gene, at position number 571, there's usually a G, and that stands for guanine, which is one of those 4 DNA bases. And actually, in this patient, that G is actually changed to an A. So that would represent adenine, which is another one of those 4 bases. So that's all that this is saying, is that at position number 571, most folks have a G, this patient has an A, and that G to A change is what is responsible for having the signs and symptoms and the diagnosis of HPP. When we think about the protein, again, that's the one that starts with the P, that lets you know that we're talking about a protein. And so similarly, this one is saying at amino acid number 191, most folks have an E, which again, in this, if we look back to the chart on the last slide, codes for something called glutamic acid. It's one of the types of an amino acid. But this patient, instead of having a glutamic acid, has a lysine. And that, just like how the G to A in the DNA name affected a patient and gave them the diagnosis of HPP, the change from glutamic acid to a lysine in the protein is the result, and that also causes HPP. So essentially, it's, are we reporting the change based on what's in the instruction manual, or are we reporting the change on what's present in the enzyme or in the chair? Now, what I think is always important to remember, like we mentioned before with variants of uncertain significance, is that the DNA and the protein information doesn't change, but the interpretation can. So it's possible that you could have a genetic change, a mutation, that was found by a lab, and one lab might say, well, we think this probably causes disease. But maybe another lab says, I'm not so sure, I tend to be more conservative, I need more data. So it is possible to get different results for the same genetic change, be it a mutation or what have you, from different labs. And so again, this is where it's really important to go back and reclassify, and it's important to be in these registries, and it's important for folks like Stephanie and I to publish on what we've seen, so that if there are those people out there who have a genetic change, who maybe the lab that they sent their testing to says, I'm not really sure, if people are able to get more information out, then maybe that'll be changed to be having a more definitive answer. Okay, so I've got some resources here question-wise. Obviously, Softbones has got a lot of good stuff on their website under their resource center. I think the fact sheets and the patient resources in particular are really helpful. I think it's great that they put these webinars up, because it's a lot of information. It's very technical, and it's hard to remember it all. So being able to reference it is great. For those of you who've not met with a genetic counselor, I hope that this was helpful, but it does not replace genetic counseling that is specifically geared towards your presentation of disease, your family's history, and your specific gene changes. So if you haven't met with a genetic counselor and you want to have that more in-depth discussion, you can go to the National Society of Genetic Counselors website. They have a feature called Find a Genetic Counselor. You can enter in your zip code and find out where genetic counselors are in your area. And then you can either reach out to them via phone or in person. And that, I think, can help give you a more personalized assessment of what's going on in your family and in your body if you're a patient. I think it's always helpful, too, to know that you can reach out to the actual lab that did your testing. So for some of you who perhaps had rheumatologists or endocrinologists who ordered the testing and they themselves struggled a little bit with interpreting the test, most of these labs have genetic counselors, so a person like me, whose complete job it is to sit in the lab and answer questions about the test and about the results of the test to either patients or the providers that ordered the testing. And so that's, I think, a really good resource. Obviously, starting with your clinical team is a good place to start. And I know that Deb and Denise have Stephanie and I's information, too, if anyone wants to reach out directly to us. But, of course, you know, start with your clinicians and get the answers you can, and if you are having trouble finding the answers that you need, then feel free to branch out. So I think that's the bulk of what we have. We're happy to answer questions. So yeah, we'll let Deb and Denise kind of monitor questions, and we'll go from there.
Thank you so much, both of you. That was great. I just, a couple questions that we get frequently, from a lot of patients are about some of these 23andMe tests, and I know that you kind of touched on that a little bit with the labs, but can we've had patients say, I've had my 23andMe testing and HPP doesn't show up. Is there something we should be doing, or is there a takeaway from that? What would be your recommendation for that type of question?
Sure. So, I mean, it's a great topic. It's in the news a lot. I feel like it's become kind of one of the, most popular, Mother's Day, Father's Day, Christmas gifts. And I think it's perfectly safe, but people have to understand what they're getting. And so medical genetics and genes that are related specifically to certain genetic conditions, a lot of times we call those single gene disorders. Sometimes they're called Mendelian genes, and that's just for the scientists who discovered them. But for those things that essentially have a single on-off switch, that's really what medical genetics is at this point really being focused on. Those are not the kinds of things that 23andMe are testing. They are more looking at those polymorphisms, like we mentioned before, to track things like potential susceptibility for hypertension. So there's not one switch for having high blood pressure. There might be 45 genes that we know have maybe a tiny % effect on whether or not somebody gets high blood pressure. But of course, their exercise level, their diet, all of those things can also affect it. No one can exercise enough to not have HPP. So they're kind of two separate categories. So 23andMe and Ancestry and those types of tests are not medical tests. They might give you some risk information about generalized things like hypertension or diabetes or where you're from, but they're not the type of test that you're having done through a clinician, through a medical laboratory.
Okay, great. Thank you for that. That helps a lot. Another question I see that came in from somebody on the call is, how do we find out where our specific mutations originate? Is there a way to find that out?
So not completely. So there are some mutations, like Stephanie mentioned, who where they tend to aggregate in certain ethnic communities. And that's, in genetics, we call that something called the founder effect. And basically all that means is that a genetic mutation happens, because they all had to happen for the first time somewhere, and they happen for the first time. Take the Japanese population, it's a good example. Until fairly recently in history, that community was very geographically isolated. So when a specific genetic mutation crops up in a community that doesn't have a lot of admixture, the frequency of that mutation actually can compound over time. And that's often why we will see that certain genetic mutations couple with certain ethnicities. With that being said, so much has now changed, and the amount of admixture has occurred that we aren't really able to track mutations specifically from an ethnic perspective. But what I think sometimes people are asking is, how do I track it through my family? And that is a different question. And so for a family with dominant disease, again, because one change is enough to cause disease, we would anticipate that if a patient has dominant disease and they inherited that non-working gene copy from their mom or their dad, we would presume that their mom or their dad, whoever gave them that copy, was also affected with dominant HPP. What's challenging in HPP is that because this can present later on, because it can be a little non-specific, as Stephanie mentioned, there's tons of misdiagnoses, we may not be able to ever go back and confirm. So unless we have the family members able and willing to have testing, that's really the only way to track through the family where these actual genetic changes came from.
Great. Ashley, I can see there's more questions, but I can't see the questions, so I don't know if you can potentially read them for me.
Of course. We had two questions come in. One of them is there a website where frequency information can be found?
So as far as mutation frequency, no, but there is a database. So there was a French researcher by the name of Dr. Mornay back, really, I would think in the 80s and 90s, who did a lot of studies on the babies with a very severe disease. And so he actually did put together all of his findings. They did genetic testing on those kiddos, and that can be Googled. So if you Google HPP mutation database and Mornay, which is M-O-R-N-E-T as in Thomas, you'll find this very, very, very large chart that basically has all of the subunits of the gene laid out and all of the different genetic mutations. There's over, I think, 400 that have been recognized. So there's a lot of them. It can be a little overwhelming for patients. That's where I would really say if you have a genetic counselor, I would ask them to take a look, or your geneticist, and look at the database and look at the specific information that's been associated with the mutation or mutations in your family. And that could help give some insight as to what HPP might look like for that family.
Yeah, and can you elaborate a little bit more on one point about that? Because, you know, we have patients who are newly diagnosed who figure out what their mutation is, and maybe it's an adult, but when they look at that database, let's say one of the patients who originally had that mutation was diagnosed as having infantile. And so people will say, well, I have infantile HPP, and I try to explain to them that may not be the case. So can you kind of elaborate a little bit more on what the classification of the diseases match with the mutation may not necessarily be how the disease manifests itself in reality and in the clinic?
Yeah, sure. So one of the biggest things I think to remember when we're looking at Dr. Mornay's database is that he was only looking at the babies with very severe disease. So these kiddos who were born with multiple fractures or who had very poorly formed ribs and chest and had very bad respiratory problems. So he was only looking at a very small section of that spectrum that Stephanie's slide showed with the big arrow going from very severe to odonto HPP. And so he was only looking at, I think, a very small section. With that being said, the vast majority, if not, I believe, all of those patients did have two mutations. And so those individuals, again, either had recessive disease or they had what we call that double dominant effect. And so what we're finding out now, 20, 30 years down the road, is that some of those parents who at that time were presumed to be unaffected carriers actually, in fact, had dominant disease. It just hadn't manifested or been problematic enough to actually have been diagnosed. So when you see that database say perinatal or infantile, often it's really important to make sure that there's a likelihood that there are actually two mutations there. And as you would imagine, if you have a second mutation, that's only going to worsen things. And so that's typically why those patients had severe disease, because they had two mutations.
Great. Okay, we have one last question. This one says, our family's mutation is referred to as the Mayflower mutation. It's been reported only 40 U.S. families have thus far been identified with this mutation, and that we're all most likely related. Would the database pinpoint this more accurately?
I don't know off the top of my head. So Dr. Mornay did most of his work in France, and he did have patients travel to him. So if someone had traveled, if a family had traveled to him for that study, it's possible, but I don't know if that's in his database. I think what's interesting about those mutations is, yes, it does mean that there's some genetic link over time, or that you have some ancestor that tracks back to, again, kind of that founder effect, wherever that first mutation was. Again, the idea being that someone who came over on the Mayflower had this specific mutation, and then it kind of spread further out. So maybe you all have a common ancestor, but it's not necessarily that you're going to be closely related or know these people by chance. I I don't think that the Mornay database is going to give you any more insight on that. I think really what we're all talking about is something that we call in genetics genotype, phenotype, correlation. And what that means, genotype means your genes, what genetic changes you have, and phenotype means the actual signs and symptoms that we see. And so what everybody wants to know is, can you look at my gene report and can you tell me what this is going to look like? And right now, we can't do a very good job of that. What we need to do is, like we said, get the data from registries, publications, all of those types of things, making sure that the labs are looking at all of these sources of information so that we can better describe that. There are genetic conditions where being able to look at the specific mutations does give us an ability to predict and make some pretty good predictions on what things will or won't look like. We'll never have a perfect crystal ball, but it can help. And I do think that we're still kind of in the infancy of that. With HPP, we're learning a lot, but we have to get that information published and we have to get it readily available so that we can move from not feeling like we can tell people very specific information to being able to help guide them a little bit more once we know what the genetic changes are. But we're still getting there.
Hey,
Great.
Deborah, a couple more questions have come through. I don't know if you still have time to answer them. If not, we will provide all these questions and chat questions to Deborah and Denise if you guys would like to follow back up. But if you have time, there's 2 more questions that came through.
I can take
Okay,
more.
Okay, go ahead.
Perfect.
I can't see them for some reason on mine.
No
so
worry, no
Ashley, if you
problem.
don't mind.
Of course. One of them is there a correlation between the type of mutation or the address of the mutation via the severity of the disease?
So there can be. So based on where the mutation falls in the gene, correlates to where in the protein the problem arises. And so there are certain areas of the protein that tend to be more vital. So a lot of times enzymes, or in this case, the protein is an enzyme, enzymes' entire job are to break things down. So if the mutation that a person has is in an area of the protein that affects its ability to break something down, that might be more severe, or we're getting more kind of into the biochemistry of it, but the short answer is yes. There are some mutations that, because of the effect, kind of like we talked about with the chair, if we don't have any legs on a chair, it doesn't function as a very good chair, versus if I put, the back on the wrong side, it's uncomfortable to sit in, but I can still sit in it. So the answer is yes, and that is some of the information that the labs do use in order to interpret whether or not specific genetic changes qualify as a disease-causing mutation, or as a variant of uncertain significance, or as a non-disease-causing entity. So yes, and we're still working on defining that better.
Great. And then we have one more that came through. If my daughter has 2 mutations, is that considered dominant or recessive?
It's a great question. And right now we're kind of still trying to define it. I would say from the biggest thing that would be important really is whether or not either of the parents are manifesting any symptoms, because we don't have that crystal ball, because we don't have that data to determine those genotype-phenotype correlations, there's really no great way of looking at a child, in this case your daughter, who has 2 mutations and saying, yes, that's absolutely autosomal recessive, or well, that's probably a double dominant effect. Now, again, we could predict somewhat based on her presentation. If we were talking about a child who had this very severe perinatal form, then that maybe leads us to think one thing, and if maybe she was a little bit older, it might lead us to think something else. But I think essentially what we would be going off of is that the risk to have another child with the same type of HPP as your existing daughter would follow that 25% rule, because that would mean that future child would have to inherit both non-working copies, one from mom and one from dad. And again, we can see some variation between siblings. It's not always exactly the same, but we can say with some decent confidence that the risk would be 25% for the same severity level as the daughter that you have now, which is why you're on the webinar. But I do think that because we're learning a lot about whether or not these recessive cases are recessive versus being double dominant, it's not a terrible idea to have both parents get a baseline screen, have an ALPL checked, maybe take a look at their history from a skeletal and dental perspective and just see if anything is lining up. Because we have seen that in the past. And unfortunately, like I said, we're still learning a lot. And so I'm hoping that in 5 years, we'll be able to answer a lot of these questions with way shorter and way clearer answers.
Great,
Terrific.
thank you. Deborah, that was all the questions that have come through so far.
Okay, great. Well, I want to thank everybody who's on the line for attending tonight and for your engagement with the questions. And again, this webinar will be made public. It's being recorded. We'll be sure to send you the link once it's available, and it will also be available on the Softbones website. But with that, we include our first in the series. And again, we will have a part 2 coming up and another webinar next month with Dr. Rush on vitamin B6. So stay tuned, and I hope that everybody has a great night and a happy HPP Awareness Day. Thanks so much.